Sustainability virtual seminar series. We are happy to offer this opportunity for those interested in computing and sustainability across the globe. This virtual seminar series is being organized by CompSusNet with support from the National Science Foundation and Cornell University. My name is Doug Fisher, and I'm the Director for Outreach, Education, Diversity, and Synthesis of CompSusNet. I and other members of the outreach team, which includes Carla Gomes, Christiane White, and Rich Bernstein, will be scheduling talks on computational sustainability regularly and posting the URL on this slide. And you can also find recordings of past talks at this URL as well. We have talks by Professor Michela Milanov, University of Bologna, on December 1, and Professor Nicole Sintoff of Ohio State University on December 15th. So tune in for those too. During the webinar, you can ask questions through the question and answer facility on Zoom, which will be related to the speaker as time allows. If you have questions that you would like to ask yourself, you can raise your hand, and we will promote you to panelist if you'd like, but we will happily ask the questions for you. If you want to, go ahead and find the chat window and chat a hello to everybody. So we can see who you are. I would suggest chatting to all panelists and attendees. CompSusNet, in addition to the web page, has several social media outlets, including Facebook, Twitter, LinkedIn, and CompSusNet blog, again, on the website. Today's speaker, it's a pleasure to introduce Professor Kristian Kersting. Professor Kersting is he's the head of machine learning in the CS department at the Technical University of Darmstadt, Germany. And it is a pleasure to have you, Kristian, and I'm going to let you take it from there.
Okay. Can you hear me this way?
Make sure that your mic is unmuted.
It should be unmuted. Can you hear me? Guys?
Yes.
Okay, so you can hear me.
Yes.
Good. Let me share the screen. Let's see. No, not that way. We had to do it this way. Okay. Come on. So I hope you can see my slides now.
Yes, see them very well.
Good. First of all, thank you for inviting. As said, my name is Kristian. If you have any questions, also feel free to send me emails afterwards or just try to stay in contact. I would be very happy. So thanks for inviting. However, I must also admit I'm super, super nervous. And that's not only because it's almost night in Germany, but I also think that you're the actual experts, and I just hope to contribute a little bit. So because I am a little bit nervous, I asked one of my friends to give the introduction so that in a kind of ice-breaking fashion, so that I am hopefully a little bit less nervous. So let's see what he is telling us about the whole story and, so to say, setting the stage.
Hi, everybody. Normally, I'd begin these remarks with a joke about data science, but about half the stuff my staff came up with was below average. But as all of you know, understanding and innovating with data has the potential to change the way we do almost anything for the better. That's why my administration's opened up massive amounts of government data to the public for the first time, with more than 135,000 data sets available for download at data.gov. Think about the weather and map apps we check every day on our phones, many of which are powered by open government data, along with countless other apps and services. Or our new precision medicine initiative, which joins data science and healthcare to accelerate treatments for disease. We want more Americans to dream up and deploy innovations like these, to solve problems, save lives, and create new jobs and opportunities. That's why I'm so pleased to welcome Dr. DJ Patel as the federal government's first chief data scientist. And that's why I'm asking you to help. As DJ likes to say, data science is a team sport. That's why we want you, America's data scientists, to join us in this effort. Help us build better digital services for the American people. Help us unleash new innovation in areas like healthcare and climate change. Help us change this country and this world for the better. Thanks.
So I think there's a lot of truth to what Obama is saying. However, it's not just an US-American problem. I think it's an issue that we are all facing, so the whole world. And I think at least Europe can learn a lot, because as far as I know, we still don't have a chief data scientist at the EU level, and definitely not at the German level. So anyhow, whatever we think about it, there's definitely this arms race to deeply understand data. And whether we like it or not, I think there will be this long-lasting trend to make informed decisions jointly based on people, data, and smart computers, whatever that exactly means. And I think in the context of computational sustainability, and that's what you all know, I guess, one could pretty fairly say that data is the new soil, and we should really consider how we can harvest this data to somehow help improving our planet, or in the context a little bit, at least in this talk, how we can help it to feed a hungry world. So why is that such an important, and yeah, unfortunately in this way, if I say it this way, why is such an exciting research? Well, if you think about, the problem is that the world population will grow. However, our resources to provide food to everyone, unfortunately, is not growing, at least if we are not putting some investment, some efforts into asking ourselves, how can we improve productivity without increasing our resources, right? We simply can't do. So the question we asked several years back is somehow, how can we make use of data science to, well, at least start addressing these kinds of questions? So if you're a data scientist, in particular like me, if you're doing machine learning, of course you go for the standard typical sales pitch, right? So you say, hey, cool, give me your data. So you have features and you have objects, and let's say it's an Excel sheet, and we're describing our objects using these features and apply some standard machine learning tool, some standard machine learning approach. So you may use some matrix factorization, you may use boosting, you may use Gaussian processes, or some deep learning, whatever you want to use. And then hopefully you get this interesting new insights back. So if you go to companies, this is really typically what they tell me. They say, Kristian, I give you my data, and then you come back and we increase revenue. It's that simple. So I'm not sure whether it's that simple, but of course I like this vision, and I like to address this very interesting and really urgent problem. So we were wondering, can we now somehow make use of data science to get phenotypic characteristics of plants, right? So it's not only interesting to look at the genes, but then ultimately these genes will lead to phenotypes, and we have to understand this phenotypes and this variation that is produced by the genes, together with the environment, together with us. So we have these big interactions going on, and it's super difficult in general to understand and to come up with insights that go beyond what you can just read off from standard data. So if you look at that, the question is it really true what I was trying to sell a little bit, that you just take your data and you apply standard machine learning and then it's done? So that was the situation actually by now about, roughly really about 7 years back. So that was a time, so I was with Fraunhofer Institute, and I moved to the university as a professor for data mining in agriculture, because they have a lot of data, very interesting data. And they were approaching me saying, look, Kristian, we have this one question here. So we have two barley plants here, and we would like to know which of them is drought stressed, so has not enough water. And we would like to do that hopefully earlier than by the naked eye. And we would like to do so also without, let me use my very unprofessional word here, without killing the plant, right? So we would like to have it in a non-invasive way. So can we do that now somehow earlier by using sensor technology? And in particular, in our case, we are looking at what is called hyperspectral imaging. So a hyperspectral image is very much like what you know from standard cameras, where you measure RGB. So you essentially measure something like 3 wavelengths, or the energy reflected or observed, depending on what type of camera you have, at 3 wavelengths. But now at with a hyperspectral camera, you're doing that, for example, with 1,000 wavelengths, all in parallel, right? So you're getting, in a sense, your standard image like here, but instead of having just 3 values here, you may have 1,000 values here. And depending on which wavelengths you are measuring, so here you see the energy reflected here in an unnormalized way, and you also see that we were not spending much work on cleaning, in a sense, the data. But then at different wavelengths, you get different insights, like typically here, which is around, what is it? I think 450 or, yeah, not really sure. You're measuring chlorophyll, right? So you're looking essentially a little bit into the plant, and you see whether it is still green or not. But you can go very much to the right, even further, like here, and you get into non-visible parts where you can measure the water content and other little insights into what's going on in the plant. So the problem there is, the first obvious problem is that this easily becomes a big data challenge. And at least for me, of course, I'm not Google, but for me, it was the first time that I understood the original definition of what big data really means, at least in a personal way, that you cannot just use your standard equipment. So the data we got was something like 10 plants over 7 measurement days. And the way we measured in the beginning, we were just using 120 wavelengths. But even then, you get already easily into billions of data entries, in a sense of data cells. And if that is not enough, so that was about a terabyte, and by that we couldn't use at least our standard machines, and then you apply standard machine learning, it's getting tricky, because even something like a stochastic gradient is not easily working anymore, because you have only linear access to your data. So I told my students, hey, let's do some stochastic gradient, and it was actually slowing down, right? And significantly, I mean, so much that you can't even see a single iteration, because it randomly jumps to a data point by going to the very first data point and then going the whole way to the data point you have selected randomly. And then you jump again somewhere, but it jumps back to the beginning, starts there reading, going to the next randomly selected data point. So you start to understand, okay, I have to use the architecture, I have to maybe massage my algorithms. And ultimately, this whole problem is not even about terabytes, but we are collaborating with a company called Lambda Tech, where you have these kinds of plant factories. So plants are on belts, and then you can measure them every half an hour or every hour or whatever you want to do with them, but you get definitely easily into petabytes a day. So it's a little bit like the CERN, but now for plants, kind of at least, right? So we have obviously this one big problem that we have too much data, and that's of course as a machine learner, you are happy, right? So you say, hey, no problem, guys. Just give me your data, I'm happy to apply it. So first step you're doing is, instead of working here with tensors or these cubes, you're trying now to change that into the standard format for machine learning, right? So we have here several different hyperspectral images, maybe of the same plant at different days, or we have it here for different plants. So what you first do is somehow you turn these cubes into the standard Excel sheets, 1 per cube, 1 per plant, 1 per measurement, and then you may stack them on top of it. So we simplify it here, and we just try to view it as a very, very big Excel sheet, data matrix, as always, right? So that was easy, and that's why I felt back then, yeah, sure, give me your data. That's very, very easy, because now I can apply whatever you want to do. And in particular, they were interested in getting latent factors out there. They wanted to get the factors that are explaining certain reactions over time. So we just applied standard matrix factorization. And because I don't know your background, let me very, very briefly touch upon matrix factorization, right? So we have a data matrix, let's call it V here, and we would like to factorize it into 2 matrices, hopefully of smaller size, W and H. So V is just a data matrix, so you have examples again per row, and the features are the columns. And now we would like to factorize it in a way that I get prototypical or explaining factor or basis vectors here. And I try to explain now, or to reconstruct each of the original examples in terms of these basis functions, right? So if you manage and if you use only a few bases, it's like a bottleneck, an information bottleneck, and that way you get some insights into what is going on in your data, right? And I guess you all know the very standard matrix factorizations like singular value decomposition or PCA. So they are particular ways, and they have used to factorize matrices, and they have used a lot because you can nicely compress your data at hand, right? By just looking at the leading eigenvectors only, so you're kicking out this eigenvectors with low eigenvalues, and by that you can see that you get good reconstructions using only a few dimensions, because your residuals showing down here are getting rather small at some point, right? So, and there are tons of other matrix factorization techniques that all fit somehow the same framework that you have just seen. So of course I was bold, and I was saying, yeah, sure, give me your data and let's apply PCA. And even if PCA is not really scaling well, there are very nice randomized approaches for that, so that you can easily scale to big data. So there were several problems. So they were not really happy about what I was proposing. And one of the first problem was they don't want to subsample, at least back then, because, you know, they send out their students, let's say 120 days per year to measure, even in the field. And so they were looking at me and saying, oh, Kristian, so you're telling me that we may have just sent them out only a few days. That's not good. They don't like this idea that I were sending them out maybe 100 days for useless work. Second, maybe you're just throwing away the single information that is giving me, well, not the Turing Award, but maybe even the Nobel Prize at some point, right? So at least that's really how they argued. But their main argument against this standard approach is the following, that if you apply PCA or SVD or most of the standard matrix factorization techniques, then you're getting a mathematical object back. Like here, you will get an eigenvector. And they were just asking me, okay, Kristian, cool, but what is the biological meaning of an eigenvector? Can you remind us of that? Yeah, and sure, I couldn't, right? It's not easy to interpret eigenvectors because you have changed your basis function or your basis systems, so you have to interpret it again. And if you're not really into mathematics, and if you have not taken the data yourself, this can be a very cumbersome problem. And next to it, you're actually solving an optimization problem over a continuous space. So you may compute things that are biologically not producible, right? I mean, maybe you find insights that can never be realized in the real world. So they were very, very skeptical. So actually, I was rather disappointed. But then after thinking a little bit about it, I got the idea that we maybe can approach the whole problem in a completely different way. So actually, this was done with two colleagues at Fraunhofer, Christian Thurau and Christian Baukake. And we were very happy because we were looking forward to a paper that was maybe Kristian, somehow a nice co-authorship. So our idea was, okay, maybe it can be driven a little bit by this idea that you can easily discuss whether a glass is half full or half empty, right? Everyone can understand what we are talking here about. And these glasses here, you can view that as a picture illustrating convex combinations, right? Because the middle one, the half full, you can now say it's either half empty or half full, right? So depending on what type of personality you are, but definitely everyone can talk about it. So our idea was, well, why not looking at the convex hull of your data? Or let's say at least a subset of the convex hull. So like here, we are looking at 3 extreme points of all your data in a sense. Here you may look at 5, at 7, you may look at the whole convex hull ultimately. And then what you can do is you can now express all the other data as convex combinations of these few what is called archetypes or extreme data points, right? For those that you haven't captured well, you may project them onto the simplex, but then you have really this nice representation on the probability simplex where you can nicely tell people, look, your data point here at hand looks a little bit like the data point you have measured and a little bit more like the other data point and not so much like the final extreme data point. So that was working very well, at least when we told them the idea. So what we were doing is we were really implementing it. So we were taking our data metrics, right? So the hyperspectral cubes turned into a data sheet. And then we were trying to compute these extreme data points, right? So kind of illustrated here. And then we were using these extreme data points, and we considered it as a kind of rubber band around our data. And we were expressing now all the other data points as convex combinations of them. So here, for example, let's say we have selected 3 different extreme signatures or archetypical signatures, and then you can describe any other signature by saying, well, it's kind of 80% like this signature and 18% like this signature and only 2% like this signature. So they were happy, we were happy. In particular, if you look then closer at other kind of data, you can see that this way you're actually showing much more variation of your data points than compared to SVD or non-negative matrix factorization or even k-means. So you see, for example, in k-means, because we are looking at averages per cluster, all the images, all the faces here look rather similar. You see some variation, but it's not as easy for all the images if you look at these 2 images. They look actually very similar, but the parts of the image of the face are slightly different. If you look at this archetypical analysis, you actually see, because we look at all these extreme points, all the variation in your data. And just to make clear, of course, you can have also robust versions of that by not looking at just the extremes, but you look at one extreme point and you take the 100 or k nearest neighbor and you average over them, or you take the most central data point in there. So you can all do that more robust. And it's even an interesting, in my opinion, theoretical research question in theory, whether in big data settings, there is actually harmful things going on with outliers. Because maybe an outlier in 1 billion data points, it's a very interesting data point. But that's maybe a different story. So far, you see, it's easy to explain what we are doing. It's easy to explain. And it shows a lot of variation, and hopefully by that captures somehow your data. So I was happy. However, then at some point, Kristian Kersting, one of my co-authors of this original work, was saying, come on, this is not true, because unfortunately, the convex hull is not easy to compute. So there, you can prove that it's per se not easy, so it scales with the dimension in a not nice way. But you can also recall the curse of dimensionality at some point, right? So at some point, it becomes simply very difficult to compute the convex hull, right? Most likely, everything is, in a sense, on the convex hull. So there is maybe still hope, because we are looking at a subset of the convex hull. And so if you look at the manifold, hopefully in a low-dimensional space, hopefully you can still get very interesting candidates. But we are then somehow bumping into this complexity wall, and how can we compute that? So we were a little bit disappointed. But then actually we came across a very interesting work that is less fashionable in machine learning, which is called distance geometry. So in a sense, it just studies sets of points when you have distances at hand only. So there's very traditional work by, for example, Fries and Vampolar and so on, which is trying to do matrix factorization very much like what you've seen. So you try to minimize what is called the Frobenius error reconstruction. So reconstruction in terms of, so that you minimize the Frobenius norm of the reconstruction. And they were actually showing that you can do an approximate a Monte Carlo approach to do that efficiently. Generally, you can also prove that it's an NP-hard problem. So it's not easy to really compute in a deterministic way the correct answer. What we were able, using distance geometry, is to come up with an algorithm that is at least locally always maximizing the volume. So that's guaranteed. You may not get the optimum value, but without going into details, you can prove that you're not doing horribly bad compared to other optimization techniques, such as archetypical analysis by Bryman and Cutler. But that's a different story, because I would like to focus here a little bit more on the applications and what you can do with that. But you can really show that you can really provide some forms of guarantees here, even in terms of the quality you're getting here. More interesting, not only is it easy to compute in a linear way now, because the algorithm is just, in a sense, computing always the next point that is furthest away from the current simplex you're spanning, in a sense. You can also do that in an online fashion. So you can do it on a data stream, right? So because you can always keep the current set of archetypes, and whenever a new data point comes in, you just check, is it inside your current simplex, or is it falling outside? And if it falls outside, you test whether you get an improvement in volume by putting that in. So you may recall or know the work on end member detection. This is very similar, but just a more efficient algorithm. But you can also easily parallelize it, because we are looking at convex combinations. You can take any subset of data, compute the results, the K archetypes on the subset, and then you combine the results of all these subsets, again, by using the same algorithm. So you can think of a kind of tree structured decomposition technique so that you can easily parallelize, so that something that was taking before in 1.5 days is going easily down on standard cluster technology to 5 minutes. I mean, back then it was 30 minutes, but by now it's below even 5 minutes. So this way we can really hope to scale, and that's why we are collaborating with the company, whether we can really get into this petabyte of data at some point. So to summarize, so far the algorithm I was kind of sketching here, which we call simplex volume maximization, it has nice features, right? So it can compute efficiently a basis vector, actually in linear running time. And as said, you can also easily parallelize it. But more importantly, because each basis vector you're finding is a real data point, it's easy to interpret already just the basis you find here. And even more interesting, if something goes wrong with the measurement, typically people say, no, it's not me, I haven't measured wrongly, it's your analysis algorithm that is doing something wrong. But here we can really say, no, it's your data point there, so check it out. So we can also, so to say, go back to those guys who are gathering the data, the biologists or the plant physiologists, and they get involved. Then they are easy to interpret, even these combinations, because they're convex combinations, right? So you're not talking about eigenvalues, and you don't have to think about the biological meaning. It is obviously there. Indeed, we may not solve now if you change the representation, the reconstruction a little bit, then we are going away from the data points, and whether that data point would really exist, that's a different question. But we are currently looking into that in a sense of a GAN network that you may now train there as well. If you want to give it a try, you can download the code with lots of other kinds of matrix factorization techniques and in Python. Give it a try. It works pretty good. And if you use it, just tell me. I would be interested to see what you have done with that. Now, let's use this technique and see whether it really helps to do plant phenotyping, right? So we got this about terabyte of data, and we run this algorithm, and we were asking for 3 archetypes. And actually, what we were doing is we were looking at 10 of these extreme points, and then we were using some statistics to extract now the most interesting 3 of them by using a form of entropy. And you will see at least some parts how that works later on. But anyhow, we found the 3 archetypical or extreme signatures here, and they make actually completely sense. So the green one here is the typical signature that you will see for a healthy spot on your plant. So you have the chlorophyll part here, so it is green, and then you have this big jump here where you go from the visible to the invisible part of the light, which is called red edge, right? Because you go here into the infrared part. So you have this jump here. And then these 2 guys here. So this brownish one is actually, because we can now check on our image, what was the pixel that is corresponding to the signature. It was a brown spot. Hopefully, or we thought at that point, kind of drying out spot on the plant, right? And then the red one, clearly that was a dead part of the plant. There was not anything going on anymore. So we were quite happy because, first of all, it makes sense. And keep in mind, this was computed by 2 computer scientists back then. that are rather dumb when it comes to plant physiology. But then when we were asking the experts, they were even saying, yeah, sure, they are mainly pigments in this area, and there's a red edge, and so on and so on. So we were quite happy. We can now do statistics, very basic statistics here. So you're essentially just counting something, how many healthy spots do you have, how many dying out spots do you have, and then you can compare, so to say, the ratio. That's what we call drought gap here. And what you can see is after, on the 6th measurement day, typically the human can easily distinguish by naked eye whether one of the barleys is drought stressed or not. And with this technology, you can now move much earlier. It's about a week. So this is the 4th measurement day, and we were measuring every 2nd to 3rd day. So you gain 1 week already by just doing a very simple technique and then counting simply how often you have seen which hyperspectral signature, right? So that's pretty good. But to be honest, I mean, data science is not just counting, and at least on that level, but we would like to have some statistical models, right? And you would like to have these statistical models for several reasons, because you may detect, but you may also use completely predict. I mean, you can predict maybe into the future, but you may also get completely different representation of the data at hand, and by that getting a little bit more insights. So how can you come up now with statistical models there? So our idea is the following. Keep in mind, what we have managed is we have represented everything in terms of these few extreme data points, right? These extreme data points, they are spanning a simplex, a probability simplex actually, right? So in a sense, each point can be viewed as one probability distribution. And so now if you want to have this distribution over the distribution, so you want to have a distribution over a simplex or on a simplex, then the very standard one is the Dirichlet, but you may also use other distributions like a log normal, or we are currently working on new methods, but we will come back to that later. So we were just using the standard Dirichlet distribution, which has some pros, some cons. But as I said, it's not so much important which one you're using to understand what we were up to, right? So you can measure now, you take your data, you have found your extreme points, you look at the reconstructions, and now per image, for example, per measurement day, you're computing your Dirichlet distribution. And why is that interesting? Well, this, first of all, you can do very quickly. And second, because you have now distributions, you can compute distances among the distributions. Very standard techniques for computing how similar these two distributions are. And because now we have distances among the plants, among the measurement days, you can do a lot of interesting things. And what we were doing here is, for example, just a very standard technique, multidimensional scaling, right? So we were wondering, can we embed our measurements into the two-dimensional space so that you can still see what is going on the physiological level, right? And what you see here is the result. So all the green ones, keep in mind, we were not providing the information whether they are healthy or not, right? But here the green traces, in a sense, they are the healthy plants, and all the red ones, they are the drought stressed and maybe even dying out plants, right? And you see that around the 3rd to 4th measurement day, they really start to behave completely different. And that's somehow interesting because it seems like we have really captured what is going on in the plant. So you may wonder why in the beginning some of the healthy, unstressed plants go very much or behave very much like the stressed plants. This is explainable. So what we have done here is we were looking at barley during the flowering period. And during the flowering period, they are anyhow getting stressed. So it's not easy for them. It's a little bit like us humans. I don't know, what is the flowering one? Maybe if we go for our wedding or whatever, it might be in a very positive way stressful, right? So you get stressed, and but you recover, and then you go onto your healthy path again. So as said, you get interesting differences. As I also said here, we showed somehow that you can really easily parallelize it on a single core back then, so about 7 years back. It was taking 1.5 days, but then if you parallelize on a rather small cluster, you can easily get it down into the minutes part. So more interesting even, we were not making huge assumption on the distribution. So if you apply standard machine learning, typically you should ask, what is my distribution on the data so that I can apply certain techniques, and particularly here in this kind of unsupervised learning technique. Right, if you currently look into deep learning, for example, this is less of an issue for now, but even there you find papers that start to wonder, do I really capture the distribution of my data here, right? So you find all these variational autoencoders, GANs, and so on. This is all about a very similar question. And so here the interesting fact is that we just use distances where we make indeed some assumption, like if you use the Euclidean distance, it's very natural for Gaussian distributions, but it can also cover some other distributions because we are looking just at the extremes. And then on this new representation, we are learning a standard distribution. So with distributions at hand, you can not only look at MDS, but you can now also turn it a little bit into metro maps. So we were motivated here a lot by Carlos Gerstrand's work on metro maps of science. So they were looking more at scientific papers and news articles. But here I was wondering whether we can come up with an easy to understand way for people that don't like Euclidean spaces. They don't like mathematics at all. And so we were trying to use the metro maps here. Yeah, Doug?
Kristen?
Yeah.
We have a question. Elizabeth asks about the quality of the images that were used.
You mean in terms of the hyperspectral quality? Oops.
Possibly. Elizabeth, you want to clarify? Yes.
Okay, so yeah, it's a good question. So now I must admit, what we always did is we asked the biologist to take the images, and of course they were investing a lot into. So you put a little white box in the background to get a reference value for all your energies you're getting there. But you saw in the beginning this very dirty data. We were actually using that. It was very so. While we were doing our research, the biologists started to understand better the camera as well. And so over the years, they look now much smoother. But that was actually good that we did it on this noisy data, because we were actually the ones that were providing feedback. We were saying, look, we are getting this very extreme data point here that looks weird because there's a jump in the energy that we cannot explain. So yes, data and how clean your data is an issue, in particular if you want to publish in the field, so to say, in the scientific field of plants. But in terms of technique, it works whether it's very noisy or not. It's rather stable. Of course, if it's completely crap your data, you will get also crap extreme points. Is that roughly or kind of answering your question?
I believe that was probably a very good answer, yes.
Good.
Okay, thanks. That was, yeah, Elizabeth's response.
Good. So how do I get back to my slides now? Do you still see my screen now?
We see your screen, but not your slides.
Yeah, let's see. Something screwed up. Okay, so, oh yeah, and there's even more. Don't do that. Okay, let's see. Can you see the slide again?
Yes, yes.
Good, good. So, and you see here, we want to even abstract even further. So in the end, you get something like everyone started at the same point. It's namely when you inoculate, for example, a biotic stress factor. So at that point, we were even moving beyond just drought stress. And then over time, you can now get these different metro lines. So you see that some of them behave in the beginning very similar, and then they split. If they cross later on again, then be careful. It doesn't mean that you can now jump from a dead tissue to a spore release or whatever, but it just tells you that at that point, the hyperspectral signature is expected to look very, very similar. As said, we were just trying to show that you can get a kind of different representation so that you can talk to stakeholders and people that are really not interested into data science per se in a hopefully more easy to understand or to discuss at least representation. So because this is all working pretty fast, of course, you can also employ that in using different systems. So what we were looking at is then next were hyperspectral camera together with a kind of microscope, right? Because we wanted to zoom in now and see how really more fine-grained details can be captured and whether we can still explain what we get there. And you can see that now the embeddings, the traces become a little bit more smooth because we also got better in taking the data. So that's again an illustration of the answer I was trying to give to the last question, right? But you can also apply it there, and it works. And you can always project it back into the RGB and into your original image, so that you can even classify other spots automatically and see what is going on. You can also use that for different kind of quantitative trait loci, so you can really see differences in the way you breed different plants, which is very important because ultimately, as I told you, we would like to also control, maybe even automatically, the way we breed plants to make them more resistant to certain forms of stress. So I will not go into details here, but you can really see different behavior in the way that the hyperspectral signatures here evolve. So here the color is showing you the day after inoculation here, right? And then you see that two different types of barley, so you have different genetic information in there, they really behave completely different. And then this one here is more resistant than this one. And just recently, we also started to understand, hey, why just plants? So we were also using it now, so to say, on humans. So we were interested in, can we use hyperspectral images to better understand how wound healing is really working for humans? And the main point here is, again, that if you use standard clustering, it's maybe much more difficult to really get the clustering right here and to understand. And when we were working with the medics here, then using a clustering based on these hyperspectral images, you were getting much cleaner, much more, well, it was making more sense to the medics here and what is going on. So I hope this will be online soon. At least it got accepted and they have the camera ready version. And I guess in another little project, we were wondering whether we can even go into the throat. So I can only encourage people, if they have a little bit of money, such a camera, I mean, they're getting cheaper, but you may spend 100,000 euros on them, but they're getting cheaper. But I can only really encourage people to make use of this additional information you're getting from hyperspectral images. And you can really observe interesting phenomena without destroying, in a sense, people. So we were also wondering whether we can use that really for diagnosing illness for people or of people. But let's see. I mean, it's just starting now. And we were just looking for other interesting applications. So, and as I told you, it's not only about describing things, but again, because we have this nice description, so we have extreme points, and now we have distributions over the data that is now residing on the probability simplex, we were wondering, can we even make predictions? So can I say that a plant will be drought stress in the near future? So how can we do that? Well, keep in mind, we have our data, right? And what we essentially did is we were turning now each image into a Dirichlet distribution, right? So each image is a Dirichlet distribution. So what you can do is now you can look at the hyperparameters, the concentration parameters of the Dirichlet here. So this is this alpha 1, 1, alpha 2, 1, alpha 1, 2, alpha 2, 2. So they are the concentration parameters. And what you can do now is you can put on top of this a log linear model or a Gaussian process that is trying to predict the concentration parameters that are then explaining the best the distribution you have seen over time. So that's a little bit like Andrew McCallum's topic models. So he was also using Dirichlet-Aggregation Regression, which is the overall system. So you're taking a log linear model to predict latent concentration parameters, which are then used to produce here, for example, the Dirichlet. He was using that for topic analysis, and we were adapting it here now to understand hyperspectral imaging and drought and stress reactions in general of plants. And so in order to train now, what you're doing is you're, in a sense, fixing your hyperparameters, the predictions, and you train now new Dirichlets. So you're changing, so to say, the predictions of your Gaussian processes. Then you fix these observed concentration parameters and you adapt your Gaussian process. And so you go on in this coordinate gradient descent fashion, or you can also call it stochastic EM. Right? And this way, you're getting now a joint system which you can use after you have observed certain data to look into the future, because you have a Gaussian process at hand. And there you can use standard multi-step ahead drought forecasting methods, right? So you can just take a standard Gaussian process, predict the next time point, and then you take the uncertainty of your Gaussian process into account and put that into the next Gaussian process prediction. And so this way you're getting not only a prediction of the mean values, but you may also get the predictive variance into the future. And that's what you can see here. And we were really, it's really, I was surprised by that, but you can really get another about 3 days that you can take earlier action now to help the plants. You may get something against your biotic stress, or you may give just water or whatever. You may wonder why this curve is going into this very specific behavior here, but keep in mind, because these are distributions, we were looking at a softmax representation internally, and that's why it will never go down completely to 0 here in our case, because we had to put, to avoid some zeros in there. Therefore, you have this a bit extra space there, and in total, you have to be between 0 and 1. But anyhow, so you can do predictions and you can employ more advanced machine learning here, right? So to summarize so far, I hope I have convinced you somehow that scalability and interpretability are really major concerns, not only of deep learning, but of machine learning in, for plant phenotyping in general, because it's an interdisciplinary endeavor here, right? And they want to understand, and they are not trusting any machine learning per se. By now, I guess they are trusting much more, and it's super interesting, and they are asking the right questions, right? They're pushing our field as well. However, now indeed, you may not only apply or go via the simplex approach, but you may also somehow try to simplify the representation of your data so that you can better understand, and you get an interaction with the plant biologists. And so our idea then was at some point, hey, why do we have to do all the strange part and we have to think about the distributions and the distances? Now, why not looking at a different way? And we call it wordification. Let me explain it to you. So our idea was, when I was talking to the plant biologists, what they were always doing is they were never interested in the digits of the energy reflected, right? They were not interested in telling me, oh, there's 99.99876% reflected. They were always looking only at 99%, 50%, 51%. So they were just interested in 100 different values. So what you can do is you can have an a priori discretization of the data you're expecting, right? So the energy levels, you're just putting a grid of 100 possible values here. And then for the wavelength, the same, right? They get anyhow averaged a little bit locally due to the camera. So they were also only interested in, okay, this is, let's say 450 nanometers and 451, right? So you get it discretized, you get a grid here. And now you can give each grid cell just the name. So we were saying, okay, this is at wavelength 251 or whatever you're measuring. It was reflecting 98% of the energy. Okay, and that way you can build up a large vocabulary, but your observations are anyhow sparse. So our idea was, hey, that's very much like documents, right? So we are viewing now each hyperspectral signature you're getting here as a kind of sentence, right? So this is one word after the other. And then one image is something like a document. And now we would like to get in a document collection, we would like to get topics, right? So the natural idea is, hey, let's use LDA or any other probabilistic topic model you know, and let's apply it there. And we know it should work somehow, right? So there's only two problems. So first of all, in the beginning, because you have still a terabyte of data, you really need something that is performing well. And to be honest, we were not having the best machine back then in Bonn. So we were wondering, how can we improve single core LDA? So we were then heavily motivated, again, around Bly and Matt Hoffman's online LDA. But there was one additional constraint. So if you just apply their approach, they ignore, of course, that what you have at hand, a single signature here, right? It is a little bit like a function. You have a smoothness, right? So if you have observed a particular word here, you're not expecting that the next word is jumping up here, right? So what we talked about the noisy or clean data, you have some expectation that you have a smooth curve here, right? So how do you get that into LDA? Well, what you can do is in LDA, you can have a look at the prior distribution they are using anyhow for the words, right? And now you replace that by a more interesting one, where you take point-wise mutual information into account. And our problem was, yeah, okay, that works, and it's a nice paper in NIPS, but it doesn't scale. It was not clear how to do that in an online fashion, and that's what we were showing then there in the UAI paper. And then applying that, we were mainly then showing in the scientific reports paper. So as you can imagine, it works very, very, very well. So here you see the topics. So now the darker, the more likely is it that you observe this kind of word here. So here you have the word at 552 and reflectance, yeah, I don't know, maybe 0.1 or 0.08, right? So that's, you see here your standard topic distribution, which you know maybe from documents, standard documents. You now use here your words, and we projected that back into the representation of a hyperspectral signature, right? And then you see standard topics like this red edge here, so that you know that in the near infrared part, you're jumping up to much higher higher reflectance values, right? And you also get the other standard things like you want to see something at the chlorophyll part, and so on and so on. And this was really, you just put your data in there, you run LDA, and you get that out. So again, we were not massaging our data. We were just saying, compute, I think that was 10 topics, and we were getting these topics out that really conform to planned physiological knowledge. So motivated by that, we were looking not only at Drought stress then, but also at biotic stress again. And again, you get something that is easy to understand, and you may even use word clouds to make it even more interesting, because you can now really give names to the topics, and then you all of a sudden see what is really going on. And so at least the supervisor of my colleague in the biology department, she was really happy, because they were not so much interested in getting always these signatures, but now a clear understanding that they can present to the stakeholders, or if you go to breeding companies, that you can explain in standard human words, right? So you really see over time how that develops. So again, even for the biotic stress, that is really conforming to planned physiological knowledge. What we are more interested right now is that with this kind of wordification, it's very obvious to ask now, hey, can we use any kind of deep learning in there? Because there are so many interesting deep networks working on language, right? So can we now apply that to understand this hyperspectral language of plants even further? So a very obvious one is, for example, word2vec, right? So you want to find a Euclidean embedding of each word, so that words that appear very often together in a context are also close by, and otherwise you're further away. And if you use word2vec, which is highly efficient, and you apply that to hyperspectral images, you get also very reasonable distances or clusters. So here you see clusters when they are very close to each other, and you see that they make really, really sense here. And so instead of using PMI, we are now working currently on using LDA together with this kind of deep word dependency model. And you may even try to learn them maybe jointly at some point. So that's pretty interesting. And what we are also working on is trying to still get interpretable models that are deep, that can talk about probability. So I don't know whether you have heard of the Sum-Product Networks, or if you look a little bit more general, this is called arithmetic circuits, going back to Adnan Darwish and Pedro Dominguez and these kinds of guys. And the idea is that you can still do tractable inference. They are easy models typically, so you can interpret them, but they are very powerful still in capturing distribution. Now, coming from LDA, the problem is that you need a multivariate distribution over Poisson random variables. And that wasn't clear how to do it. There's very interesting work on how to do that approximately, but it's getting very mathematical and it's getting very, not easy. And it's not even clear whether you can scale easily to larger amounts of data. So our idea was different. So again, we are looking at our documents and words or features. And what you're doing is you're making use now of independency tests. So we were wondering, are there any independency tests actually for Poisson random variables? And yes, if you look closer, there's exactly, at least we were only able to find one paper from the statistics community, which provide you with an independency test for Poisson random variables. And they do it by, you check whether parameters you're estimating are stable when you perturb your data. So what you can do now is you can learn two Poisson GLMs, so generalized linear models, or Poisson trees using this kind of technology then, and you can split your words. So it's a little bit like divide and conquer, right? And then on these two subsets, what you can do is, if you don't find independencies anymore, you can do clustering. And we were providing a particular way to cluster now, in a sense, multinomial Poisson distributions. It's not so important, the details, because actually this clustering is just giving you new possibilities to test for independency, to really see independencies. So actually quite often random projections, so randomly splitting, in a sense, the data into two groups works already surprisingly well. So let's not go into details, but instead of having independencies, so where you can just look at the product of the two distributions, left and right here, at these mixtures, you're looking at sums, right? They are mixture models. So you have a sum over weighted clusters. And so you keep going, alternating between independency and clustering. And by that, you're building a tree-shaped sum product computation graph, right? And it's really interesting because you get a kind of tractable, at least for some probabilistic queries, you get a tractable graphical models over Poissons or over multiple Poisson random variables. This is very exciting to me because it's not only helping in plant physiology, but also for LDA and in many other interesting applications in medicine, where you, for example, count now how many cells you have in particular tissues and so on. So Poisson and count distributions are actually much more prominent in science than in machine learning. And I think we should start working much more on count random variables there as well. Anyhow, I said what you're getting back is a kind of polynomial, a network polynomial over Poisson distributions. And that is interesting because you can apply now completely different evaluation method approaches. So you may combine that now, for example, with SMT, so SAT modulo theory approaches, to even put additional constraints in there. We were using it to compute, for example, entropy. So here, not on plant data, but on the NIPS corpus. You see mutual information between Poisson random variables, so the words, and you see that training and learning are connected to each other, learning and algorithm are connected to each other, training and neuron, but cell and training, actually, if you look closer, it's a little bit less well connected, and spikes is not directly connected to training, so it makes sense, and you can actually prove that. So the conditional distribution of a probabilistic topic model is well known to be just the product of independent Poisson random variables. So in a sense, these Poisson Sum-Product Networks, they directly give you an LDA model, or the major part of an LDA model, but now with a hierarchy of topics. So we are currently running it, and I was hoping that I can show you these results already, but we were not able to finish. But we are currently running it now on these plant data, so that you're not just getting topics, but even a hierarchy of topics. And I hope that we can capture by that in the long run dynamics a little bit better. So that's why I can only show you here now, again, NIPS. And you see really that you get topics on each level, but then topics per se can be broken into some subtopics again. So you get that for free, and still is very parallelizable as well. And just now we got informed, I'm very happy about that, that our AAAI paper got accepted, because what you can do is here, if you think about it, what we just did, we did independency tests and clustering or random splitting, and dependency splitting and dependency splitting. Now, there's very interesting work by Arthur Gretton and all these people around Cercospora that are actually providing non-parametric independency tests. And now we can build all of a sudden graphical models without knowing the statistical type of the data or the features we are looking at. So you're getting now a model that you can train without even knowing if that Poisson, if that multinomial, is that whatever. And then only in the leaves where you have to commit to a distribution, you may have to decide, and that's where we are currently collaborating with Cambridge guys together to automatically classify the statistical type. But you may also look at a histogram representation or piecewise linear representation of your distribution there and use that for inference. So what we can provide you now is a tool where you can build a graphical model, a what-if model, without even knowing that you're working with random variables and what type of random variables. So I'm rather excited about it. Yeah? Was there a question?
No, I don't believe there was a question, but we should wrap up soon.
Yeah.
I think there will be some questions.
Yeah, but I'm close to the conclusions.
Okay.
So because of these new, deeper models, we were lucky and get some new project now currently just started about applying deep learning to phenotyping using hyperspectral images. So with that, let me conclude. In my opinion, genomic data is super interesting, but I think the ultimate goal is that we should really combine phenomic and genomic data, because we have not to only understand the genes, but we also have to understand how the genes affect the phenome. But to do that, we need a lot of data. But it's not only that you need a lot of data, but these models have to be interpretable because you're working in an interdisciplinary field here. So that's very, very interesting. And to be honest, for me, the most exciting part was that I didn't have to explain to my mom anymore what I'm doing. I was just saying I'm trying to help to solve the world hunger problem, and she understood. And that is pretty exciting for a machine learner, because typically if you talk about objective function, no one cares anymore, and they don't want to listen to you anymore. As said, scalability and interpretability, is very important. But what I was most enjoying is that it's really a team sport. So it's not just that there is a machine learner helping someone. No, the machine learner is also learning a lot of new things. So that's why I was shamelessly borrowing here a phrase that Carla was coming up for computational sustainability, right? So data scientists can and should play a key role in increasing the efficiency and effectiveness of the way we manage, understand, and allocate our natural resources while enriching and transforming data mining, right? So I was adapting it a little bit to the topic I'm working on. And I hope I have not bored you to death with that. And I hope I have convinced you we can really do something together and really enrich both fields if you do this translational or interdisciplinary research. Thank you, guys.
Thank you. Are there any questions? Have you worked at all, Molly asks, have you worked at all on satellite imagery?
That's a very good point. So at the end of my career in Bonn, I was getting actually in touch with these guys, but then we were never managing. Now now that I'm in Darmstadt, we are actually starting to look into that again, but not via the university. So we are running a AI course for students from all over Germany. And there's one guy in there who wanted to look at satellite images. But it would be great if someone else could do it. I think it's a very interesting question. So in particular, or let's say in general, airborne imaging, algorithmically it works. Whether it still gives you, because of a different resolution, interesting insights, I would say yes. I think it's a natural candidate. So, but we haven't done it yet.
Okay. Molly follows up in the chat window with much more difficult with mixed pixels, however.
Mixed or unmixing? So, not sure I'm understanding the question correctly. So, as I said, what we did is it's a different way for what is called end member detection or unmixing, right? So this is highly related. We were just looking at a different algorithmic path for doing that. That is scaling better. And then putting on top of that standard machine learning, which wasn't done before. Meanwhile, there's some other interesting work. I think it was published JMLR or MLJ, I'm not sure, on Bayesian archetypical analysis. And last year or this year, ICML, there were also some interesting new approaches.
Okay. Elizabeth asks, have you looked at sensor data like soil moisture to understand stress?
Yes, that's together with other people from the plant physiology. So in parallel to our analysis, they were taking additional, let me call it now, so to say, biochemical measures, trying to conform the statements that we were making. And yes, at least on the data we were looking so far, it got always conformed. But for the same reason, we are currently pushing a lot to have both phenomic and genomic data, because I think what we ultimately would like to do is really connecting both. And that's why we were working on this, well, we called it mixed sum-product networks, so some product networks that you can learn over hybrid domains. There I can't give you already results on the biological part, but for the other one we were doing, it's going even as extreme. You know, if machine learners are, sorry for saying that, they are sometimes arrogant because we are just working with baselines. And so when we first submitted this work, we got, well, not really rejected, but people were saying, oh, you have to run some additional experiments. And I said, hey, cool, sure, give me your data, we are running the next experiment. And then our partners were looking at me saying, yeah, Kristian, we can do it, but keep in mind the next flowering period will be next year. So we have to wait for another year. Right, and I was not used to that idea. So I was learning a lot. So, but we were doing this experiment. We were even trying to go from the greenhouse, so to say, from the lab over the greenhouse into the fields, and we were showing that some these signatures we were finding, they are super robust, and you can even apply them, even if you measure them in the lab, you can apply them in the greenhouse as well as in the field. I mean, I'm not saying always, but in the experiment we were doing, because they are so extreme, they are rather robust and they're working well. And actually they're capturing other information in there. So in a blind experiment, the Jülich people were sending us data and we didn't know what they have measured. And then we were doing this simplex volume plus clustering, and we were sending back 4 clusters we were finding. And they said, oh, yeah, that makes sense, because they were looking at fertilized and not fertilized, and stressed and not stressed, or something like that. And we were finding them without even knowing that we are looking for 4 clusters. So yes, we are trying to always accompany our results with a more traditional way of showing it.
Okay, I think this will be the last question. But maybe you could show, I was going to have you stop screen sharing, but maybe you could put it on the screen with your email address so that people could follow up.
Okay. Let me see.
Norman asks, do you see the use of hyperspectral camera in field use?
It's starting. It's starting. So it takes a little bit, they were a little bit too expensive. It's getting better now. But yes, and even companies are getting more and more interested. So breeding companies. The bigger ones, so Bayer, BASF, and as far as I know, and Monsanto and all them, Syngenta, they are doing that anyhow already. Now the question is whether we can get the farmers into that. And for that, what we have to make sure is that these images are getting a little, cameras are easier to use, right? And that's what we haven't managed yet. But there's new development using LDEs. So what the hope is that you can do these measurements and analysis somewhere at a big company or at the university, and then you figure out what you really want to measure, and then you get a kind of personalized camera, which is much cheaper and much easier to use. So there's interest, people are working on that, not fully solved yet. But yes, it will happen in my opinion. So I hope you can see my email address here now.
Yes, we see it. And I want to thank you again, and let's everyone thank Kristian again.
Thank you for listening, and thank you for inviting us. And as said, if you have questions, send an email or ask Doug or Carla how to reach me. I would be happy to get in contact.
That's great. I'll probably send you a follow-up email myself. It's been a while since I've seen you.
Yeah, really a while. Thank you, guys.
Bye-bye.
Bye.